speaker is Julia Panov. She's the Associate Director at the Tauber Bioinformatics Research Center at Haifa University in Israel, where she is working on big data omics analysis, integration, and modeling. She is involved in different projects, including analysis of whole genome sequencing, single cell sequencing, proteomics, and phosphoproteomics mass spec data, as well as others. And she is now finishing her PhD studies at Dr. Kafsan's lab in neurobiology, where she has been studying molecular mechanisms of neurodevelopmental disorders using a variety of bioinformatics tools. And so Julia will speak to us today about analysis of whole genome sequence data in cancer studies, as well as analysis of single cell transcriptomic data and bulk RNA deconvolution approaches. So Julia, over to you. Okay, can you try now?
Yes, thank you. Thank you very much for the introduction.
Co-host, yep.
Thank you for the introduction, and thank you, everybody. So today I want to share with you a little bit of the ongoing projects that we are doing at the Tauber Bioinformatics Research Center, and that I have been involved in, and which have become the basis of 3 newly developed sections on the T-BioInfo Platform. So first, I want to introduce you a little bit to the Tauber Bioinformatics Research Center. So Tauber Bioinformatics, maybe I can start sharing my screen. So Tauber Bioinformatics Research Center is a research center at the University of Haifa in Israel. And we are a team of biologists, bioinformaticians, and computer scientists. And our main goal of the center is to make bioinformatics tools accessible to a wide range of researchers. So for this, we are developing and maintaining and updating this T-BioInfo platform, which you can see right now, that has a wide array of areas, omics areas that we upgrade and update. The platform has a user-friendly interface for analysis of different types of omics data. So, of course, having access to such a platform might assist in analyses. However, many times researchers coming from different areas of expertise don't know where to start or how best to utilize the tools available on the platform, even if the tools are quite easy for use and don't need extensive knowledge of algorithms that are behind the tools. So we collaborate with Pine Biotech team on development of tutorials and study materials, exemplar projects, short courses, and specializations to enable research to quickly start using the platform for their own research. So today I will be sharing with you 2 main projects that our team, Tauber team, has been working on for the past year or so, and based on which we have developed 3 new sections of the platform. And at the moment, together with Pine Biotech team, we are working on writing up the tutorials and exemplar step-by-step guides for running the pipelines that I will be speaking about on the platform. And we hope that in a few months, these sections will be open for all the T-BioInfo platform users. So the first project that I want to share with you will be a project dedicated to analysis of whole genome sequencing data that we have been involved with and in collaboration with Professor Gutkov from Roswell Comprehensive Cancer Center. So this section was tailored actually for tumor-normal pairs of samples from the same patient. However, many of these tools can be applied to a wide range of biological questions. So As you probably know, whole genome sequencing has become a go-to technology for cancer research. It allows unprecedented understanding of genome alteration at a single nucleotide level. Using whole genome sequencing technology, we can identify genome duplications, deletions, inversions, and insertions, as well as point mutations and changes from heterozygosity to homozygosity in cancer cells. So this is the section that we have been developing and working on, and it includes pipelines that allow to study all those chromosomal alterations, point mutations, driver genes, using control tumor pairs of samples. So in our project, for example, we found great variability in chromosomal instability. What you see here on the left, right here, is one of the representative samples that we have been working with. And we saw that some of the chromosomal regions have up to 10 times amplification of this region. For example, in chromosome 12 of this genome. In another type of cancer, another representative sample on the right, you can see that much less events have happened, and the chromosomal instability is not as overwhelming as on the left. However, still here we also see some duplication events and some deletion events, as in chromosome 3, for example. If we look more in details to these structural variations in different types of cancers, we found that different cancers have, of course, different structural variations. And the pipelines allow us to look at such variations as deletions, duplications, insertions, inversions, and translocations of genomic loci with really nucleotide proficiency. For example, we can see exact points where the duplication has happened. The next problem that a lot of cancer researchers are dealing with are nucleotide alterations or mutations and differences in mutation load in normal and tumor samples. And there are basically two approaches at looking at these at point mutations. We can either look at cumulative distribution of alterations compared to the reference genome. And what you see on the left is a graph, is a comparison of normal tissue from the patient, which is green. So frequencies of mutations in normal tissue. And by mutations here, I mean alterations from the reference genome and alterations from the reference genome in the tumor sample, which is represented by the red line. And we can see that there is faster accumulation of low frequency mutations in tumor. And this, of course, is in line with our understanding of clonality of tumor, meaning that tumor cells do not have similar mutations in all of the cells. And we can compare these distributions of cumulative mutations with, for example, with Kolmogorov-Smirnov statistic, which will show us the significance of difference between these two distributions. On the right, you can see a histogram of the same distributions. And again, we can see that while in normal samples, which are represented by the blue lines, we see accumulation of differences from the reference genome at about 50%, so heterozygotic alternative alleles. In tumor samples, we see this accumulation at low frequency of mutations and also at high frequency of mutations when heterozygotic alternative alleles are moved to homozygous. A different approach is, of course, to study somatic mutations in tumors. Here, the challenge is in identifying tumor purity. And of course, when we take tumor samples, they are not 100% pure. So that means that a lot of reads from tumor sample are actually coming from normal cells, which means that a lot of times it is hard to identify whether a particular alteration is actual mutation, or it can be a mistake in sequencing or mistake in alignment. So there are many algorithms that have been developed to finding somatic mutations in tumor using tumor and normal pairs of samples taken from the same tissue, from the same patient, sorry. One of the gold standards is MUTECT2 approach algorithm, which we also have used for our study. And you can see that different cancers have different loads of somatic mutations. Finally, we can also identify driver mutations or driver genes, and based on the both on the genomic location of the mutation and the neighboring sequences next to the mutation, we can discriminate between driver mutations, mutations that drive the evolution of the tumor, and between passenger mutations that appear in the evolution of the tumor. And again, you can see that we found some genes that putative driver genes of this particular cancer. And finally, all of this information, wide range of information, can be put together in one graphical representation that helps put all the information together. And if we look at two cancers, we see that there is a wide variety of signals in two different cancers. So one particular interest or recently it has been suggested as one of the drivers of tumor evolution is the somatic integrations of repetitive elements in the genome, into the genome. And as you might know, protein coding genes are just a very small portion of our genome, of the human genome and of all mammalian genome. And a big portion of the genome is actually repetitive elements that have not been studied widely before, but that have been a focus of many studies recently. They have specific challenges, computational challenges, because of their repetitive sequence nature. So these sequences are actually very similar, and they appear in many, many places in the genome. So standard approaches of aligning genomic reads onto those regions fail because all of the reads that actually come from different parts of the genome align to just several parts of the genome. And of course, also, these regions are very hard to assemble. So many times these regions are do not appear in reference genome. However, recently, it has been one of the main repetitive element that has been proven to be active not only in tumor cells, but in other disorders as well, is line 1. It is capable of retrotransposition when it is translated and can be inserted into a different genomic region. And it has been shown that usually those repetitive elements are guarded against by methylation. But in many disorders such as cancer, metabolic disorders, neurological disorders, and others, there is hypomethylation of the genome, and these repetitive elements do transpose not only themselves, but also other repetitive elements and other genomic elements. So one of our goals in this study was to study the somatic integration of these repetitive elements into the tumor, in the genomes of tumor cells. And we have developed this pipelines that allow others also to study such somatic integrations of repetitive elements. There have been several other algorithms developed recently that also try to study these repetitive elements and their integration into genome. And as I said earlier, it is computationally, it is challenging. So we cannot use standard tools that are used in for example, regular genome sequencing analyses. So here, for example, we have an approach where we use split reads, or we call them sometimes hybrid reads. When part of the read, what you see in black is the sequencing pair-end sequencing read, and we align the sequencing read to the repeat template with part of the read hanging out, and then we continue to align the reads to the reference genome. And by this way, we can identify the putative integration sites, either integration sites that are not included in the reference genome, or integration sites that were already in the reference genome. So using this approach, we found somatic sign integrations in our 2 different types of cancer. And this is somatic integration per cell. We also had matching RNA sequencing data for the matching samples. And so we compared the expression of the same sign repetitive elements with the somatic integration into the genome. And we found that the expression of these signs are mirrored in the somatic integrations into the genome. So this was our project regarding the whole genome sequencing and analysis of whole genome sequencing data. Next project that we did in collaboration with Professor Moshe Levy from Georgetown University and his lab was analysis of single nuclei RNA-seq data from COVID-19 patients. And we have developed we are developing this new T-BioInfo section that will be also available to all the users, I hope soon. As you probably know, single cell or single nuclei RNA-seq involves, first of all, single cell suspension, and then single cell sequencing when cell barcodes and UMI barcodes, which barcode actual mRNA molecules, are connected to this read. And by reading the cell barcodes and UMI barcodes, we can identify which reads belong to which cells and which molecules. And by this way, we can get an expression matrix or expression table where we have thousands of cells from different patients, for example, with, of course, with expression of thousands of genes. So we actually analyzed recently published data set from this publication from Aviv Regev team from Broad Institute. They have published data from different organs of SARS COVID-19 patients. And they also published some information about the patients. So we were interested in kidney organ, and so we analyzed the kidney data from these patients. The first actually question that we had, and perhaps it is somehow related to the talk that we heard from Professor Andino, is whether COVID-19 actually targets kidney tissue directly, or whether what we see of kidney failure is not because of the direct targeting by the SARS-2 virus. So there is conflicting evidence. There are some reports that say that coronavirus infection targets directly the kidney, and others that fail to replicate or that do not find this to be true. We looked at our data actually, well, not at our data, but at the data that we have been analyzing. And we also did not find any evidence of reads that came from SARS-CoV-2 genome. What we studied is a host response to the infection in COVID-19 patients compared to controls. And what you see here on the graph is a after batch corrections and after normalization of the data, how the different cells, which are presented as points here, and pink points represent cells coming from COVID-19 patients, and blue cells, blue points represent cells coming from control patients, how closely they are related. And we can see that there is some cells that appear to be different in COVID-19 patients compared to control patients. Now, we were especially interested in COVID-19 patients that have some comorbidity, such as obesity, diabetes, kidney disease, and so forth. And COVID-19 critical patients that do not have these comorbidities. And of course, we compared them to the control patients. And what you see here is just the same actually graph as you have seen before, just divided into those three types of patients, and also clustered So this clustering of cells was done unsupervisedly, so that we actually cannot tell at the moment which cells which cells we are looking at. We just know that cells in cluster 1, for example, transcriptionally, they are very similar to each other and are probably very different from another cluster, such as cluster 8. So in this, what right away I think is evident from this visualization is that cells from cluster 1, it seems like control patients almost have no cells in this cluster 1. And if to present a different cluster, for example, in cluster 0, we see that actually COVID-19 patients have less cells in cluster 0 than the control patients. And it can be argued whether COVID-19 patients with comorbidity have more cells or not. And we are working right now on identifying whether this is true or not. However, this does not answer one of the most important questions, and that is, which cells exactly are we talking about when we talk about this cluster 1? So there are several approaches to try to annotate the types of cells that we are looking at. And this is our attempt. And I have to say that this exercise did not go very well. And I will share with you in a minute why. But the known marker genes that we have used to identify the cell types for these clusters seem to not work very well. For example, some of the podocyte markers, one of the cell types in kidneys, are those 3 genes presented on the left. And you can see that here, the podocyte cluster actually is these markers are expressed in this part of the graph, which is here. So we can say that actually podocytes, we were able to identify which cluster belongs to the podocytes. And also with endothelial cell markers, we were quite successful in identifying this cluster as podocytes. However, what we were really hoping to see is a cluster of different clusters of different immune cells that would point to immune infiltration or infiltration of immune cells into the kidney. However, when we looked at the markers of immune cells, for example, monocyte markers, we saw that there were almost no cells that expressed these markers, and also B cell markers. We did not see almost no cells that expressed these markers. And these failings can be related to the type of data that we are investigating, which is single nuclei data, which has been shown in the past to be biased against catching immune cells, different types of immune cells, at least in kidney tissue. So perhaps...
Sorry to interrupt. We're a little bit running behind on time. So I wonder if we can wrap up in another minute or less.
Yes, I'm almost finished. Okay, thank you. So, of course, for each cluster, whether it will be a cell type or just a cluster without a specific cell type, we can identify the molecular pathways that are either activated or suppressed in COVID-19 patients compared to the controls. And also we can try to identify the differences in the cell populations between control and disease states. which we also try to do here. So I want to finally thank Tauber Bioinformatics team and our collaborators at Roosevelt Park and at Georgetown University, and special thanks to Professor Tauber for his support of our activities, and thank you for listening.